Okay, good afternoon, everybody. And we're going to conclude our section on microbial genetics by considering the very last stage of the central dogma. Remember, the central dogma says that DNA is transcribed to messenger RNA, which is translated to protein by the ribosomes. So this is the point at which we've ended up last time. We talked about the process of transcription. DNA is transcribed to messenger RNA. That transcription is the responsibility of this enzyme, which is RNA polymerase. And RNA polymerase locates the gene by binding at an area upstream of the gene called the promoter. The promoter contains information which informs the RNA polymerase of where to begin the process of transcription. RNA polymerase opens up a transcription bubble. It separates the strands of DNA, not for very far. It's only tens of bases. And on one strand, which contains the information of the gene, called the template strand, the RNA polymerase lays down a messenger RNA strand by base pairing to the information from the DNA. Notice that, as always in nucleic acids, the growth is at the 3' end, which is here. It's the 3' end of the RNA molecule. That is where we're going to add bases by base pairing to the bases here in the DNA. That process continues. The transcription bubble is closed up as soon as the RNA polymerase has passed, and the messenger RNA trails behind like this until a point is reached which serves as a terminator. And at that point, transcription ceases and the RNA polymerase falls off. So the messenger RNA now in prokaryotes is free inside the cytoplasm. In fact, as soon as the messenger RNA appears in the cytoplasm in prokaryotes, ribosomes can clamp to it and begin the process of translation. And that's what we're going to discuss today. So the messenger RNA has to be is still a nucleic acid. DNA is a nucleic acid, so is messenger RNA. Somehow or other, we have to have a link between the nucleic acid code and the resulting protein product. And proteins are made a polymer, which is made up of amino acid monomers. Nucleic acid, nucleic acid is a polymer which is made up of nucleotide monomers. So somehow or other, we've got to get from the nucleic acid to the protein, and that is the process of translation. So I'll explain first of all that the code which is lodged in DNA consists of words, and they are in 3 base units. In the DNA, we refer to those as the triplets. And each triplet during transcription is transcribed to a codon in messenger RNA. So the codons are 3 base units which comprise a word which specify certain things. Now, if we have 4 bases and we are making words of 3, we have 64 possible combinations of the bases. And those 64 are going to contain the information for specific amino acids. 61 of those codons code for amino acids. 3 of the rest, remember we have 64, 61 code for amino acids, 3 code for instructions to the process, and actually comprise codons which are called the stop codons. So If we have 61 codons coding for amino acids, those we call the sense codons. And they have to code for only 20 amino acids. So there's a lot of redundancy. In other words, one amino acid may be coded for by many different codons. It makes no difference. They may be coded for by many different codons with the same result. And this we refer to as degeneracy. We say that code is degenerate, meaning that each amino acid can be coded by several different codons. So these are all by a lot of laborious work by scientists. These, the codons have all been decoded so that we know exactly what amino acids each one codes for. And here they're placed into a table of codons. And here it is here. You'll see they're all in units of 3, like that. This is the 5' side. So whenever you designate a codon, you designate 5' da da da, 5' da da da. So here are all the what are called the sense codons, all coding for a particular amino acid. Here are the 3 stop codons, UAG, UAA, and UGA. Those are the 3 stop codons. There is one other instruction contained in the table of codons, and that is here. And this is the codon AUG. That is the only one which I expect you to remember. Okay, and that is AUG. Codes for 2 things. It codes for methionine. It's the only methionine codon, but it also serves as a start codon. Under special circumstances, we're going to see that the presence of an AUG indicates to the ribosome that it must start the process of translation. And we'll see exactly how that works in a minute. For the rest, have a look at how redundant, how degenerate the code really is. Look at this. This is leucine. And look at these, UUA, UUG, both code there. But here, CUU, CUC, CUA, CUG, these all also code for leucine. Doesn't matter. They will have the same effect. They will produce a leucine during translation. They will bring in a leucine during translation. So when you, if you're given a codon and asked, what is that codon? Let's just say I give you the codon CAA. So, well, you can chicken pick through the whole thing until you find it. And it's glycine. There it is there. CAA. How about I say CCU? There it is there. That's proline. So that's fine. You can do that. You can chicken peck through the whole thing until you find it. But there is another way to do it. If I say ACU, what I'm going to do is I'm going to look here. This is the first position. I'm going to scan up and down first position. There it is. There's A. I said ACU. So it's in this row of A's here, right? The second position, remembering it's that row, I said C. Here's the second position. So it's in this block here. And then here is the third position, U. There it is, there, ACU. That is for threonine. So it's very easy to decipher from codons what the amino acid, the corresponding amino acid. But remember, you must be able to look at the other aspects of the central dogma. If I say to you, okay, we've got threonine is 5' ACA. That is the codon. What is the DNA triplet that would produce that? okay? And remember, base pairs. So the DNA would be T, G, T. That would be the DNA triplet that produced the messenger RNA ACA. Okay, so you need to be able to swap back and forth between the different aspects. So I said before that we have to somehow move from a nucleic acid, which is a polymer of nucleotides. We have to somehow move to a protein strand, which is a polymer of amino acids. Nucleic acids talk nucleic acid. Proteins, polypeptides talk amino acid. How are we going to get them to talk to one another? How do we take a nucleic acid code and translate it now into an amino acid code, an amino acid sequence? We have to have something that can function both as a nucleic acid and has amino acid properties. And that is this little molecule here. And this is tRNA, or transfer RNA. It's an RNA molecule. Here's a simple two-dimensional diagram. And you'll remember I told you that RNA, although it's single-stranded, it can base pair to areas of its own strand. And that's what's happening here. We've got small, short helices developing here, here, and here. And this casts the molecule into a three-dimensional shape in space. And it adopts this kind of upside down L shape. And at this end here, the 3' end, which shouldn't surprise you because that's the reactive end, the 3' end is bonded to an amino acid. In every case, when a tRNA bears an amino acid like this, we say that it is charged. And this tRNA can only be charged with one specific amino acid. And when we are kind of searching for the identity of the tRNA and the amino acid, we have to go down here to this end of the tRNA and have a look. You see here it says CGU, and that would be 3' CGU. That is called an anticodon, and it will bond base pair to a specific messenger RNA codon. And the codon that it is going to bond to is GCA, 5' GCA. Now go and have a look back at the table of codons. Sorry. And let's go. We go GCA. There it is there. And it is alanine. GCA is alanine. And look here, that is the amino acid it carries. The character of the tRNA is determined by its anticodon. And that anticodon, we're going to find base pairs to a specific codon. And that is how the codon stands for alanine. The polypeptide chain is going to be produced by an interaction of the ribosomes with the messenger RNA and with transfer RNA. And the transfer RNAs are all specific to particular codons. So as codons appear along the along the messenger RNA, so specific amino acids will be brought in at that point. So let's have a look now how it actually works. We'll go step by step through the process. Here's our messenger RNA strand, 5' end there, 3' end here. And here's the AUG. Here's the very first AUG. And when I say first, what do I mean by first? Because AUG, methionine, may occur many, many times in the messenger RNA transcript. Methionine is a common amino acid in polypeptides in proteins. So how do we know which is the first? And the answer is that upstream here, not very far away, there is a sequence in the messenger RNA called the Shine-Delgado sequence. This little small ribosomal subunit first binds near the Shine-Delgado, and it then moves down the messenger RNA strand until it finds the very first AUG after the Shine-Delgado sequence. The small ribosomal particle now binds there to this, where the AUG is, and the very first transfer RNA is brought in. And here it comes here, and look at its anticodon. Its anticodon is UACY, because it has to bond here, UAC. And what does this first one carry? Well, if you look up 5' AUG, what does it code for? It codes for methionine. So this tRNA will carry in the first amino acid, which is methionine. And it now bonds, so we now have a unit of the small ribosomal particle bound here, the first tRNA bound here, and now the large ribosomal subunit comes in. And the small subunit checks before it allows anything more to happen. It checks that everything is lined up. So here's the first tRNA bonded here, UAC to AUG, carrying methionine, and it is in an area of the large subunit called the P site. It's also called the peptidyl site. And remember it as the P site. And next to it, there is a site called the A site. Look what's happened. We have our first codon, first amino acid. Next door to it here, we have an empty codon, UUA. Now, here's rule number one. The ribosome will not tolerate an empty A site with an empty codon. So as soon as that happens, a new tRNA will be brought in. Here comes the second tRNA involved. Here it comes, here it's anticodon AAU. Here it bonds here to this codon, AAU. So it's going to come and sit here in the A site. So now we'll have two tRNAs adjacent to one another. The next thing that happens is very important. And you must get firmly in your mind. And that is this methionine that's sitting here in the P site will form a peptide bond with that leucine, not the other way around. Whatever is in the P site will form a peptide bond to the newest amino acid that has been brought in. And that leaves this tRNA uncharged. Here's rule number 2 for the ribosome. The ribosome will not tolerate an uncharged tRNA sitting in the P site. Instead, the whole system moves. The specific term is translocation. The ribosome moves along the messenger RNA by one codon. It goes over one entire codon. Okay, moves one codon over. So what has happened now, we had an empty tRNA sitting here. Here's this anticodon UAC. The whole ribosome moves relative to the messenger RNA. Here it now sits in this area here, which is called the E site for exit. And as soon as it gets there, it leaves. It is kicked out. That means that a new tRNA must now come in to sit on this codon here, GGU. New tRNA has the anticodon CCA. Look up 5' GGU, and you'll see it is carrying glycine. Now remember what happens. We've now got this our little methionine leucine chain. This now bonds there. So we'll have methionine, leucine, glycine. That is the same order as these codons: methionine, leucine, glycine. That leaves this empty. Remember, this one's gone. Okay. So that one's gone. Now we affect this peptide bond here. This is now an uncharged tRNA. Ribosome won't tolerate that, kicks it out, and now it translocates, brings a new one, etc., 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 etc. It just carries on and on and on as long as these codons are coding for amino acids. As long as they are sense codons, the translation will continue to grow the polypeptide chain, step by step by step by step. Empty A site, bring in a new T, bond over, empty tRNA, move, translocate, kick it out, bring in a new one, etc., 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 etc. And this will continue along the messenger RNA strand until such time as one of those 3 stop codons is reached. And here is one here, UGA. And when UGA appear, any of those 3 stop codons, when it appears in the messenger RNA transcript, then there's no amino acid brought in. Instead, a complex protein structure is brought in called the releasing factor. So a releasing factor is brought in here, and that results in the small and large particles separating. They liberate the polypeptide chain, which goes off for processing in the cell to become a viable protein. All of the tRNAs, the uncharged tRNAs that have been released, they are recycled. There are mechanisms in the cell to recharge them with their specific amino acids in accordance with their anticodon here. And that is the whole process. So let's see if this little video works.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including...
Oh, man. Sorry, no, it's just not working. Never mind. No, sorry, it didn't work. But there is that little animation there for you. And you can watch that. It just briefly summarizes. I've given you extra detail. Just take note that, in fact, that this little video pertains to eukaryotes, but it doesn't matter. The principles of translation are exactly the same. Okay. So remember, I'm just going to flip back here quickly, all the way back here. Remember, I told you that the messenger RNA actually trails behind the RNA polymerase during transcription. In prokaryotes, as soon as that happens, as soon as messenger RNA appears inside the cytoplasm, ribosomes will grab onto it and will begin to translate. So this is a marvelous picture. If you look here, you can see here's a DNA strand there. The RNA polymerase is running along, and it's trailing behind it, the messenger RNA. And the messenger RNA, as soon as it appears, ribosomes grab onto it. So have a look here, and you'll see that here's the RNA polymerase. trailing behind, messenger RNA, which you can't see, but you can see where it is because of all of these ribosomes that are clamped onto it. So we can tell the direction that the RNA polymerase is running because this strand here, messenger RNA here, is the oldest strand. So this RNA polymerase here, wherever it is, has been on there for the longest time. It's trailed behind it. It's still making messenger RNA. And so this ribosome up here, this is the newest ribosome to attach there on this strand here. And the other ones all trailing behind. And this is called a polysome or a polyribosome. And this only occurs in prokaryotes, that we have simultaneous transcription and translation taking place. And trailing behind each ribosome, there will be the new polypeptide strand. And in actual fact, as the polypeptide strand drifts into the cytoplasm, the processes of maturation of that polypeptide strand will already begin. So it will begin folding up and receiving whatever treatment it needs to take it to being a functional protein. So in prokaryotes, this is a very, very efficient, very rapid system. Now, in the eukaryotes, it's a little bit different because don't forget the characteristic of the eukaryotes. They have a nucleus and they have a nuclear membrane around the chromosomes. So as transcription takes place, the messenger RNA is not passing into the cytoplasm, it's passing into the space of the nucleus itself, the nucleoplasm. And there are no ribosomes there. So you're not going to have the same translation system starting immediately. Instead, the messenger RNA has to be exported from the nucleus into the cytoplasm of a eukaryote. But that's not all. Eukaryote genes are organized a bit differently. They have areas of information which will be translated, and interspersed between them, they have areas of DNA which will not be translated. They may be transcribed, but they will not be translated. That which is going to be translated, those areas are called the exons. Here, here, and here in this one, but there may be many, many, many exons in a gene. Interspersed, there are areas which will not be translated, and these are the introns. Now, the introns remain somewhat mysterious. It is evident that some of them just seem to contain what is referred to as junk DNA, which has been accumulated over the eons, but it is also becoming clear that there may be important information also contained in the introns. But before translation can take place, the introns have to be clipped out. And they're taken out, in fact, by small RNA molecules acting as enzymes, which cut them out, and they splice together the exons. And if you want to remember, a key to remembering the names is the exons are expressed. They are going to be expressed as protein. And the introns are not. So all the introns are taken out. The exons are clipped together. And in eukaryotes, there's some more processing of the messenger RNA that takes place. At this end here, at the 3' end, they add many, many, many A's, adenines. It's called the poly-A tail. And at this end here, they add a little methylated cap. And that then, the mature messenger RNA is then exported through the nuclear pores into the cytoplasm where translation can begin. Okay, so, all right. So now I just very, very briefly wanted to mention to you some of the ways in which genetic control over gene expression takes place. Remembering, some genes are run off pretty much constantly. Those are also called the housekeeping genes. They are genes which produce a product which is constantly used up and constantly needed. Other genes are only turned on at very specific times, and only when the gene product is needed for a specific purpose. And then as soon as that purpose is finished, the gene is switched off. This is a very complex topic, but I'm going to show you briefly that for prokaryotes, there is a particular system of organization which allows this kind of control to happen. So first, I want to remind you that genes always have a promoter. This is an area of control, and that is the area where the RNA polymerase binds preliminary to producing a messenger RNA strand. Now, in the prokaryotes, there is a system of genetic organization where closely related genes, let's say in a particular metabolic pathway, are linked together in organization. So here we have 3 genes. They're called the structural genes. They are the genes which are going to be read off to produce a messenger RNA transcript. They share a promoter. So once that pathway is called for, then the promoter, the RNA polymerase, will read off all 3 genes. It'll produce a single messenger RNA transcript, and that needs to be processed before it is translated, before we get to the mature protein stage. But the genes linked in a pathway have a common promoter. This is called the operon structure, and it has further elements to it. First of all, here are our 3 structural genes. Here's our promoter, and then interspersed here, there's an area called the operator. And the operator is a switch. And that switch, depending on what gene you're talking about, that can be either on or off. And its on or off nature is governed by a protein produced by a regulatory gene. And that regulatory gene, although it's shown here as being close by, it may not be. This may be remote, very remote from the structure. The promoter, operator, and structural genes linked together like this is called an operon. And this is a type of organization specific to the prokaryotes. So let's have a look and see how the operon is actually operated. There are 2 principal types. The first type of operon is called an inducible operon. Now, this is a type of genetic structure which is off almost constantly until a specific circumstance arises. And this is the way it operates. Remember, we have the regulatory gene somewhere producing a protein which acts as a repressor. That repressor in the inducible operon, that repressor binds almost permanently to the operator. The promoter is upstream. And now, because this repressor is bound here, the RNA polymerase cannot proceed. It cannot proceed past here to transcribe the structural genes there. Instead, it remains stuck here until such time as something specifically releases the repressor. The repressor has to be inactivated. And very often what it is inactivated by is some substance which needs to be metabolized by these products of these genes. So in other words, a substrate for the activity of this metabolic pathway. If it binds here, it inactivates the repressor comes off, and now the RNA polymerase can read off and transcribe all of the structural genes, and the pathway becomes active, proceeds. As it proceeds and it metabolizes whatever it was that induced activity, when that's used up and it's no more, then the repressor becomes active again and switches the whole pathway off. A very, very fine-tuned way of controlling metabolic pathways so that they are only active when they're strictly needed. Otherwise, it's a waste of energy. These genes downstream here are producing enzymes which take a lot of energy to produce. If there's nothing for them to operate on, it would simply be a waste. It's much more efficient to switch the pathway off until it is needed. So these are the inducible operons, and their condition is generally off until they are switched on to do a specific task. The other kind of operon is called a repressible operon. So this type of operon runs all the time. It is on until it is specifically switched off. So it has it's still got the regulatory gene up here, it's still producing a repressor, but the repressor is inactive. And it is inactive until such time as some substance arrives which binds to the inactive repressor and switches it on. And this will switch the repressor on, and it will clamp to the operator and switch the gene off. So this is commonly seen in systems where the gene normally will metabolize something, let's say glucose, until something else appears which could be metabolized. And you don't want to metabolize the glucose anymore, but you want to metabolize the other substance. And when that happens, that's the other substance itself acts as the co-repressor, switches off this particular operon so that another one can take over. That would be a typical situation. Okay, so I'm just going to, in detail, show you the inducible operon. And this is one of the most famous of the inducible operons because it's the very first one that was ever described. And this is the lac operon. And the lac operon is, the purpose of the lac operon is to metabolize lactose. Now, lactose is not a common substrate. It's not a substrate that bacteria will see the whole time, and not all bacteria are able to metabolize it. You have already encountered one that metabolizes it extremely well, and that is E. coli. So E. coli has an inducible lac operon for those occasions when it is presented with lactose. Lactose is commonly called milk sugar. So it has the typical operon structure. Here it is. Here's the regulatory gene, somewhere away from the gene itself, somewhere away from the operon itself, rather. Here is the operon, or the promoter, an operator, and these three structural genes. And these are linked in a pathway, metabolic pathway, to metabolize glucose and take it up. Metabolize lactose, sorry, and take up the products. So these are all genes which are closely related to one another in function. So the system works because this regulatory gene continuously produces a repressor protein by the normal processes of transcription and translation, and produces a repressor protein. And that repressor protein binds at the operator. And it sits, this operator is pretty much permanently occupied by the repressor. And the promoter here is the RNA polymerase sitting on the promoter, and it cannot move. It cannot begin transcription until this is removed. but lactose or products of lactose act as an inhibitor, if you want to think of it that way, of the repressor. So we go back here, and this is our situation. Here's our repressor active, and it's sitting blocking the lac operon, and the presence of lactose that is called the inducer, and it renders the repressor inactive. So it comes off now, it's blocked from joining up with the operator, and now the RNA polymerase can transcribe all of the products. The messenger RNA and the products can be produced by translation. So this is a typical inducible operon. Realize that these things are all going to be active. They're going to metabolize lactose and allow the products to be taken up by the cell and used in different things. The concentration of lactose in the cell is going to drop, and when it gets low enough, then the repressor is again active and will again bind to the operator and stop the process. So the gene is only on while lactose is present. Okay, so to finish off, I just very, very briefly want to tell you that one of the characteristics of DNA that is essential for evolution to take place is that the sequence of DNA is not immutable. That, in fact, the genetic sequence contained in the genes is subject to mutation, which is a permanent change in the base sequence of the DNA. Now, the problem is that these mutations may be harmful. That, in fact, mutations very, very frequently, more frequently than not, mutations are harmful to the functioning of the organism. And only occasionally a beneficial mutation may arise. Now remember, this gene sequence, the base sequence in the gene, is coding for something which has a very specific function. And that function has been fine-tuned through evolution. And the chances are, if you mess with that, it is going to be harmful, but very occasionally, these changes may be beneficial. But in addition to that, as I'll show you in a minute, many of the changes, because of the degeneracy of the DNA, the genetic code, many changes are simply neutral. They have no effect whatsoever. Now, there are various ways in which mutation can occur. It can occur naturally, and that is most commonly because of errors made during replication. So, and those are called the spontaneous mutations. They just happen on their own during the normal cell processes. But other mutations may be induced by mutagens. And the mutagens are agents which cause mutation to arise. And those can be chemicals. They can also be things like radiation. Even ultraviolet radiation induces changes in genetic sequence. So we are continually subject to mutagens. We are continually exposed to them. But the genetic systems have repair mechanisms, which are very complex. I'm not going to go into them. But we do have repair mechanisms. But those repair mechanisms can be overwhelmed, or a mutation can be so recalcitrant that it's very difficult to repair. We are just going to talk briefly about a single base, or the impact of single base changes. But remember that mutations may in fact involve more than one base, but most commonly mutations occur just in a single base, and it's called a point mutation. So let's have a look here. This is not from your textbook, so this is, but this diagram is simple and it explains it perfectly well. So we're going to look at a DNA sequence, TTC, and at the mRNA level, it is that the mRNA transcript from this triplet would be AAG, that's our codon. And look up 5' AAG and you'll see it codes for lysine. Now, I said to you that very often, mutations may be neutral. We call those silent mutations as well. And that is because of the degeneracy of the code. So, for example, if I change the C to a T, and here's the mRNA, it is now AAA instead of AAG, it still codes for lysine. Lysine, let's have a look here. I'm just trying to remember AAG to AAA. Let's go. We'll say AAG here, lysine, and here's AAA, still coding for lysine. A change of G to A is silent. It is a neutral mutation. It will still produce lysine. There'll be no impact on the organism. Excuse me for this. Okay. So the next kind is a mutation, very, very dangerous mutation. And this is one which introduces a stop codon. So here we have our DNA, TTC, when it's not mutated. Here we've mutated that first T to an A. The messenger RNA now is UAG, and that is a stop codon. And if that mutation occurs in a sequence, the translation will stop. And the polypeptide will be truncated and almost certainly completely non-functional. So this has potential. This has the potential for being an extremely dangerous DNA mutation. And they do occur. There are human diseases which are associated with such so-called nonsense mutations, premature stops. But many, many, many mutations are missense mutations. And these are mutations where, for example, a single base change results in a completely different codon being introduced with a different amino acid associated with it. So here we go. Here's DNA TTC. Now we go to TCC in the triplet. In the messenger RNA, AGG. Now, instead of coding for lysine, it codes for arginine. Here's another one. If we change that T to a G, TGC, we get messenger RNA now is ACG. That is the codon for threonine. In this sequence, if we had a DNA sequence where a lysine was supposed to be there, but it has been mutated to either of these, we will get a completely different protein. That's a different protein amino acid sequence, and that may be extremely deleterious. A single point mutation like this in the gene for hemoglobin, for example, changes hemoglobin from normal hemoglobin to sickle cell hemoglobin. Sickle cell disease actually arises because of a single base change, a single base mutation. And it has all the massive effects that impact somebody who has sickle cell disease. Okay, now there's just one other. This is from your text, so you can look this up. There's one other impact of mutation, and that is when we don't change a base, but we take one out entirely, remembering that our sequence starts at a specific place. Translation begins at a specific place, and that is the first AUG after the Shine-Delgado sequence. So here's the first one here. We begin with methionine. Here's our normal, this is just demonstration one, but we say methionine, lysine, phenylalanine, glycine. Then there is a stop codon there. So this is our 1, 2, 3, 4, 5, 15 base sequence for this little mRNA strand. Now just imagine this triplet here in the DNA, A, which normally produces a U codon, which codes for phenylalanine. We're going to actually take out entirely that A. And it would also require that we take out its corresponding T on the other DNA strand. But that can happen. It can happen. So we're going to mutate this by taking out the A on the template strand. Now what happens? Well, now our entire sequence is thrown out. We still begin at the AUG. Here's our first one, AUG. Here's our second codon there, AAG, still the same. But look at our third codon. Now instead of U, we have U, G. And that codes for an entirely different amino acid. What about our next one? Now we have GCU instead of GGC because we've shifted. We've taken out a base and we've shifted the entire sequence back one base. And this is called a frame shift. And it is probably the most, apart from the nonsense mutation, this is one of the most dangerous of all mutations because it invalidates the whole sequence. From the point at which the frame shift, the base is removed down, the entire sequence shifts in a frame shift. And now we have an entirely different sequence, entirely new codons all the way down until a stop codon is reached. A similar impact would happen if we would take out 2. We would frame shift by 2 and have an entirely new codon sequence. If I take out 3, which theoretically can happen, if I take out an entire codon, I simply miss that codon. Everything else will stay in sequence, but that entire codon would be missing. But if I only take out 1 or I take out 2 bases from a codon, I induce a frame shift, which any of those, removing 1, 2, or 3 bases from a codon can have massive impacts on the sequence thereafter. Okay. And so I'll leave it there. I will be writing to you later today, and I'll see you again next week.